with Aprami and Rachel. Please go ahead.
Sure, thanks. I would love to. So hi, everybody. I'm really excited to introduce to you our next two speakers, Aprami and Rachel. They are both students of my online course on science communication for early career scientists. This is a 6-week online program that included students from across the country and international working on various projects related to IARPC. The purpose is to not only include more early career researchers in the work of IARPC, but also to give these researchers some training in a field that they don't often get training in and they're really hungry for, which is science communication. And by science communication, I mean just translating your research so it's appropriate for various audiences. So they learn not only to communicate their research to the public, but to scientists from other disciplines, or to more clearly articulate their research to scientists in their own discipline. So today they're practicing sort of interdisciplinary communication within the marine ecosystems collaboration team. And I think you'll find what they're doing is really exciting. So first we have Aprami, and she, as she mentioned in her introduction, is a postdoctoral associate at the University of Calgary. And her research is about organic geochemistry of the oceans, but it also relates to Arctic shipping, which is a topic that comes up quite a bit on this team. So Aprami, if you're ready, you can share your screen and don't forget to unmute yourself.
Thank you, Jessica, for the introduction. Really appreciate that. Good morning, everybody. It's wonderful to be here. Like Jessica said, I'm Aprami, and today I'm here to talk about the work that I'm doing to better understand the oil spill response development in the Arctic. So before I start, the question is, why do we even need to study this? So to answer that question, we need to look at what has currently been happening in the Arctic. Over the year, the ice cover in the Arctic has been decreasing, reducing the multi-year ice cover that exists. This contributes to an increased traffic of the ships, both commercially as well as industrially. This, therefore, contributes to an increased risk of accidental oil spills within the Arctic. This increased risk of the oil spill is the reason that I'm here today talking about what I am doing. So if there is an oil spill, there are definitely response methods in play. So let's look at what are the common response methods that exist to deal with any oil spill that happens in a marine setting. One of the very first methods is mechanical skimming, which includes using devices to literally skim off the oil from the surface of the water and then collect and retain it. While this method is great and useful, in the Arctic, it cannot be applied in the same way that it can be done in a warmer climate like the Gulf of Mexico, because of the increased response time to mobilize this equipment, considering the remoteness of Arctic, as well as the increased cost of labor and equipment to get it there in the first place. So this rules out mechanical skimming. Let's look at the next one, the use of chemical dispersants. Chemical dispersants have been used several times in spills, especially in the Deepwater Horizon spill of 2010 in the Gulf of Mexico. We saw it used tons and tons to break down the oil into smaller droplets so that they disperse and emulsify in the water far more quickly. This, however, cannot be used in the Arctic because the most likely oil to spill there is the bunker oil, which is the one that is used by the ships while they're trafficking goods or customers across the port. Another problem is that the use of these chemical dispersants is banned in Canadian waters, ruling them out as a response method. Moving on to the third one is in situ burning. In situ burning has been practiced several times to get rid of the oil that gets spilled, leaving behind nothing. This, however, becomes difficult when the water is surrounded with ice, as well as The use of bunker oil, like I previously mentioned, results in the spillage of bunker oil, which is a heavier oil and does not light on fire as easily as something like gasoline or diesel would, ruling it out as a likely option to be used. Finally, we come on to the fourth method, which is microbial degradation. So microbial degradation entails the use of the microorganisms that are already resident in the water column and eat up these hydrocarbons that get released accidentally in the marine ecosystem. So this sounds pretty good. It's natural, it's breaking them down. How about we look into that further? When the oil gets released into the environment, the hydrocarbons are surrounded by the hydrocarbon degrading bacteria, almost forming a ring around the oil droplet. They then result in breaking the oil down into water and carbon dioxide by oxidizing what is present of the hydrocarbon. So what this results is that your oil disappears, gets converted to water and carbon dioxide, which is relatively clean. And because these microorganisms are inherent, there is no additional additives that need to be introduced into the environment, and therefore no repercussions to deal with the adverse effects of these things. However, to see if this actually works, and if this works, how fast would it work if a spill would occur, we need a lab. So because it's not allowed to legally spill oil and then observe it and see how it goes away for a long time, we need to find a natural lab that exists. The way around it is finding an oil seep. So an oil seep is produced when you have a reservoir below the Earth's surface, which gets pressurized but has nowhere to go. So the small cracks develop on the surface, which results in small volumes of oil constantly seeping into the marine environment. Because this introduction of the oil occurs at such a slow rate, the microorganisms in the environment have time to adapt to that setting, and therefore are more used to using these hydrocarbons as a food source. These are the microorganisms that we are trying to target as the natural cleaners during the case of an oil spill. So to do this, we developed a sampling plan where we go ahead, get these microorganisms out and see who they are and how efficiently do they work. For this, we ended up doing some of the dirty work in the Arctic. The first challenge was to select the sampling site. Where did we go? We went to Baffin Bay. We went to a place where there's already a lot of petroleum hydrocarbon potential that has already been documented. And within the Baffin Bay, we chose the site Scott Inlet, which has a very active petroleum system. There have been several spills that have been reported within this place starting from 1976. There have been instances of oil even coating the carbonates that have gone offshore. So we know that this is a site where oil is present, and because oil has been present for such a long time, we expect to see microorganisms that actually feed on this oil and have already gotten accustomed to this kind of environment. Once we figured this out, the next step was to actually go there and look at what things look like at the bottom of the ocean. What we observed was that the bottom of the ocean was actually very rocky, which made collecting sediment cores, which we would have traditionally done, impossible without breaking the core. Instead, we ended up using a remote operated vehicle to go down and scoop up some good sediment and to get that back into our labs. The interesting bit was we were able to find a seep site. We were able to find a site where we could see the oil bubbling out. We could see the gas bubbles bubbling out. And we also saw these fluorescent white colored mats, which we assumed to be the bacterial mats within that area. So because we had the ROV and significant amount of ship time, we were able to scoop up all that microbial goodness and get that back into our labs. The next step was to look at what are the good things that are present at the bottom of the ocean. What we ended up observing was lots of corals, and small organisms that were present. However, the plant and animal population at this site was significantly smaller than any other sites that we had sampled during that trip away from a seep. Beyond just collecting the sediments, we also ended up looking at some of the water. We used Niskin bottles to collect the water from the bottom to the top to be able to look at some of the DNA and the RNA that might be present in the water because of the microbes that inherently live in that water column. We also extracted the water for dissolved organic matter to be analyzed using a Fourier transform ion cyclotron resonance mass spectrometer to look at what are the biomolecules that are present in the water. So what the information is present in the dissolved organics has a huge capacity to give us information about where did the water come from, which were the organic sources that fed it, how long was that feeding, was it organic, was it from an anthropogenic source, was it terrestrial? So all these things give us a huge chunk of information about essentially a history of every single thing that has happened and occurred in the water column. Once we have the water, the next step is to look at some of the preliminary results. This slide gives you an idea of what was our sampling transect, and I'll walk you through why did we do what we did. Station 0 was the place where we found the site of the spill, where we saw the oil bubbling, we saw the methane bubbles coming out. We decided to sample in a concentric circle around a 1 kilometer mark and then a 5 kilometer mark. And the idea behind this is to get an idea if the sediments and the water are in any way connected at the site of the oil seep and further away, to get an idea how long does it take for the microorganisms that are resident there to get accustomed to that kind of setting, and if these microorganisms are specialized, or if it's the same microorganisms who are present everywhere and they just choose a different feeding option. Once we collected all these samples, we measured the dissolved methane and the dissolved hydrocarbons in these samples at all these places. What we observed was that at Station 0 and the site of the spill, the concentration of methane was highest in the bottom waters and gradually decreased from about 60 to about 10 nanomolars, which is a substantial decrease for the methane, indicating that it's the methane that is getting saturated from the oil seep, and as it's going up and up, it's just getting more and more dilute. For the hydrocarbons, we observed that as we were going away from Station 0, which was the site that was identified as the oil seep, the amount of hydrocarbons that were dissolved in the water reduced, and also the concentration of the hydrocarbons was highest in the deep waters and decreased as we moved up to the surface. And this result specifically was taken using the FTIC-RMS. The next step was to look at some of the microbial communities. As a part of the preliminary observations, what we ended up looking at was just the bottom waters at Station 0, at 1 kilometer, and at 5 kilometers away. And what we observed was that the microorganisms at Station 0 in the bottom waters were actually different than the ones that were 5 kilometers away, as well as a kilometer away, which shows that there's definitely something special. There's definitely something, a feast going on at the case of the spill where very special guests are invited. So the next step is to identify these special guests and characterize them. So this gives us a segue into the next presentation part, which is what are the experiments that are planned based on the information we currently have. The next step is to set up microcosms using the water and the sediment samples that we have collected. Excuse me. A microcosm is a smaller environment that you're setting up away from the marine ecosystem. And the idea behind this is you're giving the microbes the water, the sediments, the home that they're used to, but in a more controlled lab setting where you can actually see what is happening. This is going to be done for Station 0 as well as 1 kilometer and 5 kilometers away. We will then be introducing a little bit of oil to these mini environments to see how quickly do the microorganisms at the site of the oil seep break that down, and how quickly do the microorganisms that are actually at a site that is 5 kilometers away get accustomed to this new food source. This experimental framework would give us an idea into characterizing the microbial and organic baselines in the natural oil seeps in the Arctic area, as well as give an idea of the genetic mapping of what kind of organisms are present, who are these special guests that have been invited to this feast of the oil. And using this, we're essentially getting closer to identifying if bioremediation using microbial degradation is a viable option as an oil spill response.
Oh, that's okay. It doesn't bother me.
And better be able to inform decision-making and policy development processes. With that, I come to the end of my presentation. I would really like to thank every single person who's on the call, and especially Jessica and the people who've organized this program. It was an absolute adventure to be a part of it and learning a lot from it. I do believe that the process, the interdisciplinary stuff, the multidisciplinary stuff is the way to go when approaching an environment so unique and so pristine as the Arctic. At this point, I'd welcome any questions if we have time for some. Thank you.
Thank you so much, Aprami. That was a great and fascinating presentation. And welcome to the Marine Ecosystems Collaboration Team, by the way.
Thank you.
Are there any questions? Sure. Are there any questions for Aprami?
Hi, this is Sarah. It's not really a, oh, did somebody have a question? Oh, Aprami, this is Sarah. I am just wondering if you've had any interaction or connection with the Arctic Domain Awareness Center program run at the University of Alaska at Fairbanks, who's also doing a lot of oil spill response work?
Not currently. We're very new to this. We just ended up getting these samples last year in the summer. Before this, we were only working towards the background baseline mapping. It's only now that we're looking into the oil spill response. So we're just looking into that, but I am aware of their work, and in the future, if any collaboration is possible, we would definitely welcome that.
Great, I'll try and make a connection for you. Thank you.
That'll be great, thank you.
Any other questions for Aprami? Thank you so much, Aprami, and I hope to see you around in these meetings. And also, if you will have new developments or results that you want to share, talk to Jessica, and we can schedule a follow-up presentation in the future. Thank you.
Thank you.
Next is Rachel. Rachel, can you hear me?
Yeah, I can hear you.
Go ahead. Thank you.
All right. Okay, can everyone see that okay? All right. Yes, yeah, my name is Rachel Lekanoff. I'll be talking about my research into phytoplankton and their microdiversity in the Bering and Chukchi seas today. First, I'd like to acknowledge my co-advisors, Andrew McDonald and Eric Collins. We are all at the College of Fisheries and Ocean Sciences at the University of Alaska Fairbanks. So before I dive into the phytoplankton, I'm going to cover a few basics just to get us all on the same page. As I'm sure we all know, the oceans are imperative for the regulation of climate change. They absorb a lot of carbon from the atmosphere, and then the phytoplankton in the ocean take that carbon, or the carbon dioxide, and produce half of the world's oxygen supply. Of course, that happens in the Bering and Chukchi seas as well, and those regions are especially known for their productive waters, in part driven by the phytoplankton. There are strong seasonal blooms of phytoplankton that support a rich benthic community, and that productivity also supports organisms at higher trophic levels, such as marine mammals like whales and other fish and seabirds and people. So another unique feature of my study region in the Bering and Chukchi is the physics of the sea. So we know that there's multiple converging water masses driven by different currents throughout the region. It creates a very dynamic environment for the whole ecosystem. And in light of rapid climate change, which is disproportionately affecting the Arctic more than other regions, it's really important to understand the phytoplankton dynamics and community structure that goes on here so we can better understand how it might change in the future and how that might affect further on up the trophic levels. So with that background in mind, that brings me to my driving questions for my research. And I want to know who are the dominant taxonomic groups and how much microdiversity there is, and then what drives the structure of the phytoplankton in the Bering and Chukchi seas. Is it like the nutrient, the temperature, salinity content of the water, or is it other biological factors? So my objective one, that begs another question: what is microdiversity? So for the purpose of my study, I am defining microdiversity as the number of ESVs within species. So ESV stands for exact sequence variants. They can be thought of as unique strains within species. And they're also sometimes called ASVs in the literature, apochon sequence variants. But basically ESVs are defined by a 1 nucleotide difference in DNA. So within our DNA ladder with one of these, and we zoom up further into on a molecular level, one difference in like an adenine or a guanine that counts as a different ESV. And these are similar to maybe a more familiar way of doing this, which is operational taxonomic units, or OTUs. Those have traditionally been used in genetic studies to define taxonomic groups. So these ESVs that have one big advantage over OTUs is they can be directly compared to other data sets that are defined by ESVs. So if you generate OTUs, they're not necessarily comparable to other data sets of OTUs. So ESVs are a great way to make the information more open and accessible to other researchers. So I'll be using ESVs to explore the relationship of taxonomic groups within the environment. And so by using ESVs, we're hoping to see just how diverse the phytoplankton community is in the Bering and Chukchi. All right, so where did I get my data? On this map, I've shown the black dots are all the stations that we visited. We took part in 2 different cruises in the summer of 2017. In June, we took part in Asgard, which UAF was one of the bigger contributors to that one. Asgard is Arctic shelf growth, advection, respiration, and deposition rate measurements. And then we also took part in Ambon, which was in August of 2017, and that was the Arctic Marine Biodiversity Observing Network. So Asgard is in red, Ambon in blue. So we did the usual sampling approach for collecting phytoplankton, which is get water from CTD, filter it. In this case, we filtered it onto mostly 0.2 micron Steravex filters, pictured there on the right. At each station, we got about 1 to 5 liters of seawater, depending on how much biomass there was that clogged the filter. We got the surface and bottom depths, and usually did every 10 meters throughout the water column, which the water column is maybe about 50 meters on most spots. And if it was present, we hit the chlorophyll maximum and the thermocline or pycnocline or any other oceanographically interesting feature. So after collecting all these samples in fieldwork, we take them back to the lab where we extract, amplify, and sequence the DNA. As I mentioned earlier, we defined the sequences of DNA using ESVs. So those are the taxonomic groups, the subspecies. So getting into the results, I used these data sets of phytoplankton ESVs to address my first objective, who are the dominant phytoplankton. As it turns out, only 3 orders of diatoms are needed to capture the diversity, and these guys are in all caps: the Bacillariophyceae, Mediophyceae, and Rhizosolanaceae. And then in the columns here, it's a little hard to see, so I'll zoom in. So I picked out the big 3 genera that could be used, or most well represented in the data set. As we can see here, those are the Nichia, Catastris, and Placiosauria. So these guys are all large cell chain-forming diatoms. They're very highly productive. Because they're so large, they can also supply a lot of biomass to the benthic floor to support the community down there. And as you can see in between the 2 groups, so a lot of the other ESVs, so the subspecies here, there weren't as many. So the Nichia, Catastris, and Placiosauria, they all had really high counts of ESVs. So that means that we saw a lot of variety within those genera, which was kind of interesting. So now that we can see who the big players are, I also took a look at what variables might explain why those organisms were there. So I took a look at a few different variables. So these are all CTD plots generated from CTD hydrographic data and nutrient data that were collected along this line on Ambon. So this is one of the DBO lines. On the plots we've got on the y-axis we have pressure or depth, and then on the x-axis is the longitude. So if you work your way from left to right on a plot, it's taking you from offshore to further inshore towards the coast. Okay. And so because we know that phytoplankton blooms are affected by stratification and other physical parameters, we're expecting to see these communities change a little bit, maybe over the course of the different water conditions. So over offshore, we have more mixed or stratified layers. Inshore, they're getting more mixed. But it turns out that these communities, they didn't really respond a whole lot into these changing conditions. So in order to investigate that, I did a constrained correspondence analysis on taxonomic groups and these environmental variables as explanatory variables for each sampling site. And very little of the variability was explained by these variables, maybe less than 10% for most of them. So another avenue we are currently exploring is looking into more biological influences on these organisms. So we're thinking that the ESV, so the different taxonomic groups, may be better explanatory drivers of community structure instead of looking at nutrients or temperature salinity. So I'm still working on that. Hopefully I'll have more results in the next few months as I wrap up my master's thesis. So some takeaways from my studies so far is there is a lot of diversity within phytoplankton. On this talk, I focused mostly on just the diatoms, but I also did a similar analysis for chlorophytes, which are a clade of microgreen algae, and they also behave similarly. There wasn't a whole lot of environmental drivers that explained their abundance or their community structure. So likewise, we'll probably do another similar analysis on that. We'll look at the taxonomic units or taxonomic groupings as explanatory variables of their distribution. So the reason that we care about this diversity within the species is our thinking is that with more diversity, these taxonomic groups will have more resilience in face of climate change. So hopefully all this diversity will mean a little more robust communities in face of a changing ocean. So future work, my project here is sort of step one in a project that one of my advisors has had in mind. So our goal is to couple this phytoplankton data set and explore the interactions with bacterial communities. Since we know that phytoplankton and bacteria do interact and it has implications for productivity and the carbon cycle, so we want to see just how sensitive these bacteria are to selecting phytoplankton hosts to the KRILL family level, genus level, or ESV level. And this is a fun little picture my advisor Eric made. So each colored cluster here corresponds to a different phytoplankton group. And it's just an interesting way to lay out and see how differently or how these communities are related to one another across a broad range of genetic data. So, and you can follow more project updates here on, Eric does a lot of updates on Twitter, so there's a lot of updates on the project there. So Twitter handle if you want to check it out. And yeah, with that, I'd like to acknowledge my funders, especially my native corporations, Alaska Corporation, the Aleut Foundation, the Calnungan Tribe. They supported a lot of my scholarships throughout undergrad and now graduate school. And of course, all of these funders for supporting our project. So especially NOAA, the NOP, NPRB, NSF, and CFAR. So thank you.
Thank you so much, Rachel. Rachel, a great presentation. Are there any questions for her? I have a question, Rachel. You mentioned diversity in phytoplankton in one of your last slides. How stable is that diversity from year to year? Can you see the same level of diversity on an annual basis?
I'm not sure actually. So I only have data from 1 year so far. We do have samples from 2018, but none of them have been processed yet, as far as I know. So that's an interesting question, but I wouldn't be able to say just off of my data set so far.
So maybe you can compare with some publications from over the last few years, but it will be interesting to see how those species or different types of plankton evolve or not over time. Thank you.
Yeah.
Are there any other questions? If not, we'll move on to the next presentation by Karin Ashjian from Woods Hole Oceanographic Institution. The title of her presentation is The Impact of Shelf Break Upwelling on Beaufort Shelf Distributions of Zooplankton and Fish. Karen, go ahead.
Okay, thanks, Guillermo. Thank you, everybody, for listening to me today. And it's a tough act to follow, those two great presentations. So I'll be talking about an ongoing project that my colleagues and I are doing. We're working on the Beaufort Shelf, as the title says, and I've listed my co-PIs and colleagues on the first slide. The project is primarily funded by the National Science Foundation, but we also have contributions from BOEM, particularly looking at the seabird distributions, as well as actually the USGS, who's supporting one of my colleagues. So I've talked about this project before, but I thought I would just show this a few slides to remind you of what we're trying to do. We are motivated by the observation that you usually see beluga whales along the slope in the Beaufort Sea. And in this diagram from Sue Moore, it's old data from aerial surveys, but it's still a very striking graphic. The yellow dots are the belugas and the red dots are the bowhead whales. And you can see that they really are differentiated spatially in where you can see them. So the belugas are along the slope. And we wanted to know why. And we hypothesized that they're found there because they can find high numbers of their Arctic cod prey at that location. We further hypothesized that the upwelling along the shelf break would concentrate copepods and krill, which are the prey of the Arctic cod, that would then attract the Arctic cod to feed there. And then the cod would be present in high numbers so that they would provide a good food source for the whales. So we were looking at this food chain. So our study area is indicated by the red box. It was located more than 50 nautical miles to the east of Utqiagvik, sort of due north of Nuiqsut, but quite far away from there as well, and also way to the west of Kaktovik. And I'm going to show some highlights from the we've had 2 field sampling years. I'm going to show some highlights. This isn't going to be a comprehensive examination of what we found, but just some of the more interesting things that we found in the last few months of our analyses. So we did 2 cruises on the research vessel Sekuliak. The first one was in 2017 during the early fall, and the second one was during August of 2018 in late summer, when we should have seen different sea ice conditions during those 2 years. And indeed, we did see very different sea ice conditions. In 2017, we didn't see any sea ice. And in 2018, we saw a lot of sea ice. In fact, I think 2018 was a little bit remarkable even during these times for this portion of the Beaufort Sea. And these satellite images show the sea ice cover at the beginning of our cruise on the left in 2018 and at the end of our cruise on the right in 2018. So you can see our box was sort of located at the nexus of lingering sea ice in the Beaufort Sea. And it was interesting to follow this throughout the remainder of the fall season, in part because we had 2 moorings sitting underneath that little smudge you see on the right that we were unable to recover. And nobody else was able to get back either because of the sea ice. And so they're still sitting there waiting patiently for us to come back and get them, hopefully this summer. So as I said, we did two cruises, and in these figures on the top figure, you see the cruise tracks and the station locations for each of the cruises, with 2017 on the left and 2018 on the right. I want to point out the very different cruise tracks you see between the 2 years. We have nice straight lines in 2017 and not so much in 2018. And also our spatial coverage was a lot less in 2018 than in 2017. And this was because of the extensive sea ice cover. So we couldn't sample along our straight lines, couldn't sample along all of our lines, and couldn't go as far to the east as we wanted. And you can see our attempt to go out and get our moorings if you look at that little squiggle going along the slope in 2018. We also sampled along several of these lines a number of times. So what we would do is come into the area and sample along these lines under different upwelling conditions. And so what I'm going to show in the next few slides is data collected along this line during both of the years. And what we looked at was hydrography and currents, and then zooplankton and fish abundances from net samples. We also collected data on backscatterers from the acoustic instrument mounted on the centerboard of the Sekuliak. We did marine mammal and bird observations, and then again, we had a mooring component to the program. And also, I'm going to show you data from a mooring. If you can see it pop in, there was a mooring located right in the middle of our study area. So looking first to see how much upwelling we had during the 2 years, we experienced very different wind conditions. We had, in 2017, there were 2 periods of nice, strong upwelling winds separated by a period of non-upwelling winds, so relaxation of the shelf break upwelling. However, in 2018, we had very few upwelling winds. Most of the time, the wind was mostly from the west, which is in fact a downwelling winds, and that shading is indicating the upwelling winds in these figures. So we had 1 day of upwelling winds in 2018, except at the very end of our study period, when we had all of a sudden upwelling winds showed up. But unfortunately, at that time, we had to leave the area because we had to get back down to Nome. So these different upwelling conditions meant that we had different hydrographic structures. And so looking at these plots, these are showing the mean sections for salinity and temperature across that transect that I highlighted when we were looking at the cruise tracks. You can see, particularly in the salinity and the left graph of the leftmost graph, that in 2018, the isopleths were sloping upwards along the slope. Look particularly at the 34, which is that really dark red one, compared to 2018 when they were sloping downwards. And this is indicative of upwelling ongoing, or in the mean in 2017, but not in 2018. On the right panel of this figure, we've identified the different water masses that were present based on temperature and salinity characteristics. And you can see we had 5 different types of water masses present. I want to point out that in 2018, we had more Alaska coastal water due to the persistent west winds. Winds coming from the west apparently pushed the Alaskan coastal water along. And we also, in both years, had the winter water at about 100 to 150 meters. And notice that off the slope in the deeper water, we had Pacific halocline Atlantic water. And in 2017, you can see how that's been crawling up the slope due to the ongoing upwelling. We can look at some of our mooring records to understand the time of the temporal response of the ocean to the upwelling winds. And this just shows a snippet of data collected using the mooring that I highlighted on the cruise track from 2017. So the dates are along the horizontal axis, and then the lovely colors show the directions of the current in the bottom graph. The very top line that you see here is wind speed. This is east wind speed, so when it's above that horizontal line, we've got upwelling conditions. And then the middle line is showing the salinity. So when upwelling occurs, you get more saline water and the Atlantic water coming up the slope. So you can see that when upwelling begins, as shown is the solid vertical line, you start to see the wind increasing, then you see the salinity starts to increase as the more saline water comes crawling up the slope to the mooring location. In the bottom panel, you see that there's a reversal in the currents at about 60 meters. And this is the long slope jet. When upwelling is not occurring, it goes to the east, and under upwelling conditions, it reverses and goes to the west. And this happened twice. And what's interesting is that it took about a day for the ocean to really respond to the onset of the upwelling winds. Now I'm going to show you data we collected using the acoustic instrument, the EK60, that was mounted on the centerboard of the research vessel Zooplankton. I'm showing you data from the 38 kilohertz echo sounder. And what we did was we calculated volume backscatter, and then we identified it as being from either cod or krill-like scatterers. And from that, we could identify regions where we saw krill or cod backscatter, and then schools of cod or krill backscatter. And schools is a really tight, high-intensity aggregation of one of those types of scatterers. And so as an example, the top row shows just the volume backscatter for cod. Then the middle graph is showing you the regions where cod were seen, and this is the more diffuse cod-like backscatter, and then the bottom shows schools of cod, which are those really cohesive aggregations. We found there was a persistent, very high, very cohesive patch of Arctic cod along the slope at about 200 to primarily on 200 to 600 bottom depth contours. So on the left, you see the geographic distributions of where we saw these schools of the different types of scatterers, and the cod are shown as the cyan. And you can see it's located at 200 to 600 meters. It's further offshore than the dark blue dots, which are where we saw the krill schools. So those were found at shallower depths. On the right panel, I've plotted this in a different way. I plotted longitude on the horizontal axis, and on the vertical axis, I plotted the bottom depth at which the different schools were seen. This is not the depth in the water, but the depth of the ocean floor. And again, you see the cyan schools are the cod, and they're found at deeper bottom depths as you go from west to east than the krill schools are, which are the dark blue lines. Except when you go further, interestingly enough, further out to the east, we did have some vagrant krill schools out at some of the deeper water that may have been associated perhaps with some kind of physical feature. So I then took these data and I plotted them in a different manner. This is all the data, and this is again the schools. And so the bottom depth at which each school was observed is on the horizontal axis, and the depth in the water column at which each school was observed is on the vertical axis. And krill are in dark blue and cod are in cyan. I've put these two boxes on here to show that how the two differentiate this space. So the cod schools were found at deeper depths from say 200 meters or deeper bottom depth, so further out in the slope, 200 meters or deeper primarily. And the krill were found at shallower bottom depths, so higher up the slope. You can see that looking at the horizontal axis. They were spatially segregated in the water column as well. The krill school depths were significantly shallower than the cod school depths. So that they're not only are they spatially segregated along the slope, but also vertically in the water column. And these differences, if you look at the means, were significant. And we looked then to see if what water masses they were found. And here we're looking at that same mean section and on the left-hand panel. And I've put those same squares to indicate the along shelf locations and the vertical water column depths at which each of those schools were seen. And you can see that the cod, again, are found further along the slope, primarily in the Pacific halocline water or Atlantic water, and the krill were found both shallower and further up the slope in Pacific halocline water, but also into the Chukchi summer water and the winter water. And the right panel shows a temperature salinity diagram, color-coded according to krill and cod, and this is where we saw the different schools, and you can see the two of them are occupying different portions of the TS diagram. Just since we were looking for upwelling, I also plotted that one of the plots, but color coding it by upwelling and downwelling conditions, and I didn't see any obvious impact of upwelling versus downwelling on either the depths of the schools or the bottom depths at which they were seen, which was was kind of disappointing, so somewhat surprising. We also, this krill school was really persistent and it was really strong, but we noticed we did a diel study where we sat on top of one of the schools from about 1900 to 0900, and that's basically starting during periods of daytime and then going through darkness again till dawn and then into daytime again the next day. And that echogram from that diel study is shown on the left, and you can see that there is a very strong school at the beginning or during the daylight, but then during the periods of darkness, which is in the middle, it becomes much more diffuse and rises higher in the water column. And so on the right, I've plotted as a function of the hour of day, the mean depth of the different regions of where cod were found. So when I looked for diel vertical migration in the school data, which is the really high aggregations, I couldn't see it. And this is because I believe of how the schools become less aggregated. So we really didn't have very many schools during the night, and there really weren't any schools during the night because they weren't so concentrated anymore. And the school algorithm doesn't pick up this more diffuse pattern, which is seen in the middle of the echogram on the right. But the regions in the water column where we see cod did pick it up, and that's what's shown on the right-hand panel. So you do see that the cod actually were found higher in the water column during nighttime. But again, interestingly enough, of course, krill also like to vertically migrate, and so this plot shows as a function of the hour of the day, the mean depth for the cod and the regions where cod were seen and the regions where krill were seen. And you can see that again, they're spatially segregated. It would have been nice if the cod had zoomed all the way up to where the krill worked so that they were eating them during the periods of night, but that wasn't what we have seen to be happening. Keep in mind, however, that these cod might be making individual little migrations up to get something to eat in the upper water column, but they don't go up en masse to do that, it seems. And one of the graduate students who's been working on the project, Justin Suka, has been looking at cod diet, and that's what's seen here. He did see what he thinks was an upwelling signal in their diet, where the blue is what they were eating during upwelling, and the red is what they were eating without upwelling. And that was particularly pronounced for Calanus glacialis, which is a pretty large copepod and a nice juicy fat pill for these Arctic cod. But they were eating large copepods, krill, and amphipods. And krill was just not as big of a component as the diet as we had thought. So we saw very different environmental conditions in the 2 years, and we're still working on 2018 to figure out what the difference is in the ecology of the region. And it's important to note that the timing of the 2 cruises is also going to have an impact on the ecology, which led to the different environmental conditions. And I want to just say that we actually had planned to have these cruises at different times because we wanted to look for that variability. We saw great upwelling events in 2017, but very few in 2018. But we did see that we could see a one-day lag in the physical response to upwelling. And the big surprise to me was that krill and cod did not seem to be spatially coincident, either along the slope or in their depth in the water, in the water column, and that they occupied different water masses. We saw diel vertical migration. We did see that the cod were consuming krill, but that large copepods and amphipods may be more important. But these krill patches that we saw, or schools that we saw along the slope, were really, really prominent. And this should provide a favorable feeding environment for the belugas, as we had hypothesized. But it doesn't seem that availability of krill is driving the formation of those patches. And so I'd like to acknowledge everybody who's been working on this project, the captains and crews of the Skuliak, the marine technicians, the other members of the science team, the bird observers, and the gang at the Seward Marine Center, and the folks at the UA University of Alaska Northwest campus in Nome. And I just want to point out again that the research is supported by the NSF and BOEM supporting the bird observations. So thank you. Does anybody have any questions?
Thank you, Karen. Questions for Karen?
Yes, Karen, do you know when is their reproduction time? Do they have like an early and late season time for the cod?
I believe that they reproduce in the spring. And so one thing we were seeing between the 2 cruises is that we were seeing smaller cod during the 2018 cruise because we were about a month earlier. But stay tuned for that story.
Okay.
I'm sorry my light went out, but there you go.
Karen, I have a question.
Sure.
Your presentation reminds me of something we found studying upwelling in California back in the day in my previous life. We found that using modeling experiments, we found that there were 3 types of upwelling, and each type they were different because of a number of factors, but the main finding was that they were coming from 3 different depths, depending on a number of conditions on the wind, water mass is present, and so on. I believe your project has a modeling component. I wonder if the differences you found already in the upwelling events might be linked to those upwellings being, those events, particular events being originated in different depths in that particular location of the Beaufort Sea.
We haven't looked for that specifically in the physical modeling data. Just looking, we have repeat transects. So we didn't really see any upwelling events in 2018. At least we see it in the modeling data, but we haven't finished analyzing that. But in 2017, we have repeat transects. And for the 2 different upwelling events, they don't seem to come from different depths. You can see the isopleths go upwards from this, basically the same thing. In fact, we've picked an isopleth and used it to its position where it intersects the slope. As it'll crawl up and back, and we can use that as an indicator of this, how far the upwelling has progressed. So I don't think we're going to see that necessarily. I suppose though, if you had really strong upwelling winds for a really long period of time, you would start to get even deeper water coming up. But I think it always initiates from the same depths. Does that answer your question?
Sure. Yes. Thank you. I can send you one of the papers I have in mind, but your presentation reminded me of those modeling experiments. Thank you.
Thank you.
Any other questions for Karin? Thank you again, Karin. That was a great update, and I will update the website with some of the information, or with a summary of what you provided. This is relevant to at least a couple of the performance elements in the 5-year plan. I guess we are a few minutes past the hour. Are there any last-minute comments or questions before we close? Our next meeting is on May 22nd, same time. Thank you, everyone, and I will see you then.
Thank you.
Bye.
Bye.
Bye.